Welcome to the lecture series in food microbiology. In the last couple of lectures in this session, we have been discussing about the various foodborne illnesses caused by bacteria, viruses, etc. In continuation with the same theme, today we are going to discuss about two major pathogens, a foodborne gastroenteritis caused by two major pathogens, Yersinia as well as Campylobacter. Yersinia gastroenteritis is also called as yersiniosis. The major foodborne pathogen here is Yersinia enterocolitica. This is one of the three species of the genus Yersinia recognized as a human pathogen. Yersinia enterocolitica causes predominantly a gastroenteritis, while Yersinia pseudotuberculosis is associated mainly with mesenteric adenitis. But when you consider the societal impact, the diseases caused by either the enterocolitica or the pseudotuberculosis is very pale into insignificance, because the other organism in this genus, Yersinia pestis, is the causative organism for the bubonic plague. The genus Yersinia is named after the French bacteriologist Alexandre Yersin, who in 1894 first described this organism as responsible for the bubonic plague. In the genus Yersinia, which belongs to the family Enterobacteriaceae, 11 species and 5 biovars have been recognized, and this includes Yersinia pestis, the cause of bubonic plague. The species of primary interest as a causative organism of foodborne illness is Yersinia enterocolitica. This gram-negative rot is somewhat unique in that it is motile below 30 degrees, but not at 37, which means that it loses the capacity to move around once the temperature raises at around 37. But below 30 degrees, it is capable of moving. It produces colonies which are 1 millimeter or less on nutrient agar. When you consider its biochemical characteristics, it is oxidase negative. It can ferment glucose, but this glucose fermentation might not produce gas, or if it produces, it will be very less gas. It lacks the enzyme phenylalanine deaminase. It is urease positive, and among almost all the pathogens, it has a specific place because it is psychotrophic. So all these are the biochemical parameters which we use to identify Yersinia enterocolitica, that is oxidase negative, ferments glucose with little or no gas production, does not have phenylalanine deaminase, it is urease positive, and is psychotrophic. Foodborne yersiniosis was first confirmed in US in 1976 following an outbreak among a large number of school children from the consumption of chocolate milk contaminated with Yersinia enterocolitica. When you see the natural habitat, it can be isolated from variety of environmental sources, including soil, fresh water, and intestinal tract of many animals. So it is widespread, it can occur anywhere. And surveys have found that the organism in numerous foods, including milk and dairy products, meats, especially pork, poultry, fish, shellfish, fruits, and vegetables. So since it can occur in a wide range of environment as well as in a wide variety of food products, care has to be taken while processing as well as for storage so that the food does not become contaminated with Yersinia enterocolitica. The two most important aspects both about the organism as well as the disease is that Yersinia enterocolitica, it is psychotroph. So psychotrophs can grow at 0 degrees, which means refrigeration, and even in the freezer temperature. And the symptoms include a sharp abdominal pain with fever, mimicking an appendicitis. So this characteristic that refrigerator food generally can be safe, that cannot hold true here, because being a psychotroph, it can grow at 0 degrees. And The symptom can mimic appendicitis, which can give rise to a wrong diagnosis. Escherichia pseudotuberculosis is quite common in animals, and this has also been isolated in food. The strains grow between 0 and 44 degrees with an optimum growth at 25 to 29 degrees centigrade. And the growth occurs in milk and raw meat at 1 degrees, but at a slower rate. So even though the optimal temperature is between 25 and 29, it still has a range of temperature where it can survive and grow 0 to 44. But even at 1 degrees, even though the growth is slow, it can still multiply. And it can grow in 5% NaCl, meaning alkaline. It can grow in alkaline conditions and at a pH at above 4.6. The cells are sensitive to pasteurization. Illness caused by Escherichia enterocolitica occurs when commonly in children under 7 years old. This is a self-limiting enterocolitis with an incubation period of 1 to 11 days and lasting for about 5 to 14 days. Although in many cases it may persist for considerably longer duration, meaning normally it will run due course and slowly people will recover. And symptoms are predominantly abdominal pain with diarrhea accompanied by wild fever. Vomiting is normally rare. The ingested cells of the pathogenic Yersinia, which survive passage through the stomach acid, adhere to the mucosal cells of the Peyer's patches. So once the food has been consumed, which has contaminated with Yersinia enterocolitica, once it has passed through the stomach where it survives the acidic environment, it will go and adhere to the Peyer's cells, mucosal cells of the Peyer's patches. And for this adhesion, that is for this sticking together to the mucosal cells, you have certain proteins which are present on the outer bacterial membrane, and these proteins in turn, the genes for coding these proteins are coded by a plasmid which the bacteria contains. So you have plasmid encoded membrane proteins on the outer bacterial surface which help in attaching the bacterium to the mucosal cells. And this is a characteristic common to all pathogenic Yersinia enterocolitica. The adhered cell is taken up by the epithelial cell by endocytosis, where it survives without significant multiplication and it can exert cytotoxic activity. See, once the cell has got itself attached to the mucosa or by the epithelial cell, now it would be endocytosed by the epithelial cell. So it will go into the epithelial cell, it will now enter the cytoplasm of the epithelial cell. And inside the cytoplasm of the epithelial cell, it can survive. It needn't multiply, but it will survive, it will live. And its position there can be cytotoxic to the cell concerned. If it is released into the lamina propria, it will invade the phagocytic cells and then will multiply extracellularly, producing a local inflammatory response. It can also invade the phagocytic cells and then it will multiply on the extracellular environment. This multiplication in turn will provoke an inflammatory response in the body. Damage to the absorptive epithelial surface results in malabsorption, meaning because it is the epithelial lining which normally absorbs the nutrients from the digested food. So when the lining is damaged, the absorption will be affected. So this in turn will result in malnutrition, which is because the required nutrients are no longer being the epithelial cell is not able to absorb the present nutrients in the digested food because of the damage caused to it. As a result, osmotic fluid loss is characterized by diarrhea. So when the epithelial cell is damaged, one aspect is the malabsorption. The second aspect is the membrane potential is lost. So from inside the cytosol, the cytoplasm water and the electrolytes will start coming out into the stomach, resulting in the diarrhea. A heat-stable enterotoxin, which is 9,000 to 9,700 kilodalton, is produced by Yersinia enterocolitica. But what is its role in the pathogenesis is not clear as of yet. The enterotoxin produced by Yersinia is similar to E. coli ST immunologically and in its ability to induce fluid accumulation in the ligated ileal loops and to stimulate the guanylate cyclase activity similar to the E. coli ST. The Yersinia enterocolitica strains are found in environment. Hence, it is common for many of the foodstuffs to have the organism concerned. It has been isolated from raw milk, from processed dairy products, from raw and improperly cooked meats, from fresh vegetables, and in improperly chlorinated water, which means that the source of contamination can be wide. Foods normally implicated in yersiniosis include raw and pasteurized milk, ice cream, and improperly cooked meats. So if you see the flow chart of mode of transmission of Yersinia enterocolitica, there is one common or transmitted usual transmission mode versus a rare transmission mode. In the common transmission mode, we talk about the contaminated food and untreated water which has been contaminated with animal fecus. Contaminated food can contain either raw or undercooked food where the cooking has not been proper, or the food which has been exposed to a bacterial carrier. A person who has the disease handles the food concerned. Then the other option would be infected animals, from pet or domestic animals to human, infants, children. That is one aspect. In the occasional or the very rare transmitted mode, you have two categories here. From infected animals, which can be pets, the domestic animals, from young children who don't know or enough about the sanitary or the hygienic conditions to be precautions to be taken. Then human, where you have the bacterial carrier or the blood donors with diarrheal history, to humans who are immune compromised people. And the other option would be direct inoculation from the environmental sources. For example, you have a break in the finger or a small scratch in the finger which is leaving your skin open, and you are handling an environment or a vegetable which has been contaminated or a food which has been contaminated, the organism can directly enter your body. So that is a very, very rare transmission mode. Infection is diagnosed by isolating the organism from blood, from lymph nodes or tissues on blood agar or McConkey's agar. The most commonly used enrichment media are the phosphate buffered saline, which is PBS, or trypsin soya broth, TSB, and the incubation is mostly done at 41 degrees centigrade for 21 days. There can be different modes of transmission for Yersinia enterocolitica, as we saw. In the case of foodborne, it has been isolated from many food including beef, pork, liquid eggs, soft cheese, raw milk, pasteurized milk, chocolate milk, turkey, powdered milk, lentils, edible soybeans, tofu, etc. And the human yersiniosis is primarily acquired through the gastrointestinal tract as a result of the ingestion of the contaminated food, and usually raw and inadequately cooked meat or pork. The second case is human to human transmission, that is person to person transmission. This is a very rare case. The common case is foodborne transmission. The third category is animal to human transmission and water transmission. The Yersinia enterocolitica infection occurs after direct or indirect contact with infected animals. The last category is direct transmission, and in such a case, it can very rarely cause the extraintestinal disease, because enterocolitica mainly causes abnormality or infects the GI tract, causing enteric disease. So extraintestinal disease is very rare. In the case of an extraintestinal disease, the direct transmission is proposed as the mode of transmission of this classically enteric pathogen. The blood transfusion associated transmission. This can be transmitted through contaminated blood, and then it can cause what we call as a post-transfusion sepsis. based on the occurrence of the plasmid, which carries the genes coding for the surface proteins, which makes the organism adhere to the epithelial cells. We have two major types, what we call as a virulent biotype and a historical, the non-virulent biotypes. One is called as, among the non-virulent biotype, we have what we call as biotype 1A. This is ubiquitous, normally seen in the environment. And this biotype A, it is typed by, or it can be diagnosed by REP, Barr-Eric or PZR fingerprinting. And this is divided into two types, clonal group, two clonal types, clonal group A, which is the pathogenic strain, and the clonal group B, which is the non-pathogenic strain. So, among the non-virulent biotypes, which is biotype A, which have been typed by either PZR fingerprinting or the REP or the ERIC, we have two clones, a pathogenic strain and a clonal group B, which is the non-pathogenic strain. Among the virulent biotypes, we have two major categories, one which is highly virulent, the other which is weakly virulent. The biotype 1B belongs to the highly virulent group. This is also called as the mouse lethal group because almost all the mice infected with it, or you make the mice infected with it, they die. So it is mouse lethal group. This biotype 1B is predominant in North America, and this is otherwise called as the New World strains. Then you have the second category, biotypes 2 to 5, which are called as weakly virulent biotypes. They are unable to kill mice, and they predominate in Europe and Japan, which are called as the Old World strains. So two categories, biotype 1B and biotypes 2 to 5. One is highly virulent, the other one is weakly virulent. The highly virulent ones are classified as the New World strains. The weakly virulent strains we classify as the Old World strains. Again, when we look into the non-virulent biotypes, you can also classify them, type them by FAFLP, which we see later. This is again classified into biotype 1A, non-pathogenic biotype 1A. So, based on the presence of the plasmid itself, Yersinia enterocolitica can be subdivided into various biovars or biotypes. Yersinia enterocolitica biotypes are classified into three distinct groups. A historically defined non-pathogenic group called as the biogroup 1A, a weakly pathogenic group that is unable to kill mice, which we call as the non-lethal to mice, biogroups 2 to 5, and a highly pathogenic mouse lethal group, which is called as the biogroup 1B. Three major molecular techniques are normally used to type the Yersinia enterocolitica. The biotype 1A strains, these get clustered into two clonal groups, A group and the B group, when we type them by a technique which is called as repetitive extragenic palindrome, otherwise called as REP, and the enterobacterial repetitive intragenic consensus, which is otherwise called as ERIC, PZR fingerprinting. And they are again divided into two groups when typed by another technique, which is fluorescent amplified fragment length polymorphism, otherwise called as the FAFLP. Yersinia enterocolitica, it is sensitive to many antibiotics, including the aminoglycosides, chloramphenicol, quinolones, tetracycline, etc. So you have a range of medications, antibiotics, which can be prescribed to treat the gastrointestinal disease caused by enterocolitica. Next enteric disease which we are going to discuss is campylobacteriosis. Prior to 1970s, the campylobacters were known primarily to veterinary microbiologists as organisms that caused spontaneous abortions in cattle and sheep and as the cause of other animal pathologies. So human Campylobacter as a human pathogen was not known earlier. It was only after the 1970s that these, along with infecting animals, this can also cause human diseases was known. Campylobacter jejuni and Campylobacter coli are considered the most common causative agents of human diarrheal disease in many countries worldwide. Campylobacter jejuni. This is a slender, it is a spirally curved rod that possesses a single polar flagellum at one or both ends of the cell. So characteristic is a polar flagellum, single polar flagellum, either at one end or both ends of the cell. It is a slender, spirally curved rod. Biochemically, it is oxidase positive, catalase positive. It cannot grow in the presence of 3.5% sodium chloride or at 25 degrees centigrade. It is microaerophilic, requires very small amounts of oxygen, 3 to 6% for growth. So these are the distinguishing factors: oxidase positive, catalase positive, cannot grow in the presence of 3.5% sodium chloride, microaerophile, requires presence of small amounts of oxygen. Campylobacter laridis, Campylobacter concisus, and Campylobacter hyointestinalis. They have also been isolated occasionally from patients with diarrhea. Campylobacter pylori. Now it is reclassified as Helicobacter pylori. It has been associated with gastritis and stomach and duodenal ulcers. All the Campylobacter species grow at 37 degrees centigrade. Campylobacter jejuni and Campylobacter coli have an optima at 42 to 45 degrees centigrade, but they cannot survive cooking or pasteurization temperatures. So even though they can grow at normal body temperature 37 or at an optimum of 42 to 45 degrees centigrade, well-cooked food, they cannot withstand the cooking temperature or even the pasteurization temperatures. And their viability normally decline during chill or frozen storage, but they can persist under these conditions for prolonged periods, meaning even though the viability will reduce when frozen or when refrigerated, they still are alive and they can be alive for prolonged periods. And the survival has been recorded in milk and water at 4 degrees centigrade for several weeks storage and in frozen poultry after several months. So unless properly processed or cooked, they would still be alive, and they can, even in refrigerated food, you can still see the organisms. And even on storage, that is even on long keeping, it will still be present in the. They are sensitive to many environmental parameters, including oxygen, sodium chloride, about 2.5%, low pH, that is pH below 5, temperature below 30 degrees centigrade, heat, pasteurization and drying. Campylobacter jejuni has a thermolabile enterotoxin, thermolabile meaning sensitive to heat, enterotoxin that is responsible for the enteric disease symptoms. This toxin cross-reacts with cholera toxin, and the toxin production trait is plasmid linked. So the characteristic of Campylobacter jejuni enterotoxin is that 1, it is heat sensitive, 2, it can cross-react with cholera toxin, 3, the gene for the production of the toxin is in the plasmid, so it is plasmid linked. In addition, the strains produce an invasive factor that enables the cells to invade and establish in epithelial cells in both the small and the large intestines in humans. So, when ingested, it can, and if it is started growing in the gut, it can produce the enterotoxin, number 1. 2, if live cells enter the gut, then they have a factor, they can produce certain factors which are called as invasive factors. These invasive factors will help these cells to invade and establish in the epithelial. So they will go and invade the epithelial cells lining the gut, and once inside the gut, they will establish themselves there, not only in the small intestine but also in the large intestine. The main symptoms are enteric and include abdominal cramps, profuse diarrhea, nausea and vomiting. The other symptoms include fever, headache and chills. And in some cases, bloody diarrhea has been reported. And an individual can have a relapse of the symptom after a short interval. The foods can be contaminated directly with fecal material from animals or infected humans, or indirectly from sewage and contaminated water. Campylobacter jejuni has been isolated at a very high frequency from raw meat, beef, lamb, pork, chicken, etc., milk, eggs, vegetables, mushrooms, and clams, which means that you have a wide range of food which can likely be contaminated with Campylobacter jejuni. And it is difficult to control the access of Campylobacter jejuni to raw food, particularly foods of animal origin. However, proper sanitation can be used to reduce its load in raw food during production, processing, and future handling. So what happens is that since it is prevalent in wide range of food, especially animal-based food, you cannot avoid contamination. It is very difficult. But you can ensure proper sanitation methodology as well as the processing technologies so that the contamination of food by live organisms can be prevented. And anyway, the toxin is heat labile, so proper cooking and processing technology will destroy the Campylobacter produced enterotoxin. Preventing consumption of raw food of animal origin, heat treatment of the food when possible, and preventing post-heat contamination are important to control campylobacteriosis. Contamination of vegetables can be controlled by not using animal fecus as a fertilizer and by not using contaminated water to wash the vegetables. So the questions posed here would be, what would be the steps you take to prevent contamination of Campylobacter species in processed food? You have to remember not only that a wide range of food materials can be contaminated, and both in the case of Yersinia enterocolitica and Campylobacter, the preservation technology has to be different for both because one produces a heat labile enterotoxin, the other one, the methodology is totally different.